Good morning, everyone. So my name is Francesca Ronchi, and I'm a senior scientist at the University of Bern in the Department of Biomedical Research. And I'm grateful to the Bing Institute, so the BioStime Institute of Nutrition and Care of Geneva, to funding my project, which is aimed to understand the role of microbiota in brain homeostasis and development during adulthood and early life. So we are colonized by microbes on all our mucosal surfaces, on our skin, in the nasal and the mouth mucosa, in the gastrointestinal tract, in the lung, and in the female genital tract. And as you can see here from this picture, the oral cavity, which is the start of our gastrointestinal tract, finishing with the colon, are the two sites where we have the highest diversity of microbes that lives together with us in symbiosis. And here actually they show in this paper that actually the colon in human is the place where there's the highest concentration of bacteria, 10 to the 12 cells per gram of content. And this is the high concentration of bacteria that we can find on our planet even. So when we start to get exposed to these bacteria, this happens at the time of birth. And mammals, they are developing in uterus, which is in a sterile environment, even though some metabolites or bacteria products can pass through the placenta and actually already in utero prime the immune system or the organ development in the fetus. But really at the time of birth, there's the first exposure to the external environment when there's a huge amount of microbes. And there are several factors, natal factors or neonatal factors or postnatal factors that are impacting the composition of the microbiota and how we encounter the microbiota. This is quite important because in this early phase in life, which in humans is around 2 to 3 years of life, the microbes are changing quite a lot before getting to a stable level. And every factors can have a huge and a strong impact in that. So that's why lots of people are studying this first phase of development of the human beings or the animals and together with the development of the microbes, because they are developing together. Why we started that? It's because microbiota it's linked to health. So as you can see here from this scheme, the bacteria have been shown to have a role for the modulation of bone mass density, promotion of fat storage or angiogenesis, protection from epithelial injuries or pathogens. And they help us to digest undigestible food, which are essential for our health. They help us to metabolize drugs or to synthesize essential vitamins or amino acids. And for what we, in my groups, we are really interested in that, is to understand how bacteria shapes and educate the immune system, but also the neural system. So why we are interested in that? It's because from experimental models that people try to disturb and perturbate the microbiota at different mucosal sites. In our case, at the intestine, but this happens also for the lungs or the skin. And if we perturb in several different ways the microbial composition, people show that they have been observed some perturbation at the immune system, at the enteric nervous system, enteric immune nervous system, and at the central neuroimmune system. And as you can see here, there will be alteration in the composition and in the function of different types of immune cells and of cells that are in the brain, for example, like the microglia. So the microbes are really strictly linked to organ development and organ physiology. And not only microbes in adult life, but also microbes in early life phases, and even in the fetal, during gestation, in the fetal life. In fact, colleagues from the University of Bern, from the group of Andrew McPherson, show that actually the maternal microbial products, together with maternal antibodies, can pass through the placenta and reach the fetus. And they also can pass and get secreted into the milk and reach the neonates through breastfeeding. And these pathways, these components, mediate pathways that they induce the alteration of the immune system of the offspring, immune system that sits into the intestine. In fact, they show that the maternal microbiota induced an increase in certain immune cell population in the intestine, but also to an increase in production of antimicrobial peptides, which defend our organisms from pathogens or infections, and increase the mucus production. So this was very important because it was the first times where they really tested experimentally this during pregnancy, early phase, post-birth. So in my small group, we are trying to understand how the microbiota impacts the brain development, and both in adult life, but also early in life. And why we're interested in that, it's because there have been several groups working both on healthy condition or abnormal condition. And in healthy condition, we have a normal gut microbiota, a normal gut function, healthy gut function, healthy level of immune cells and immune mediators, and a normal behavior and unhealthy central nervous system function. However, when the bacteria are altered and there's an alteration of their composition and in their function, this is linked usually to increased inflammation in the intestine and abnormal gut function, but also linked to abnormal central nervous system function, alteration of the cognitive or mood and behaviors. So why this is happening and how this can happen, how bacteria can signal and can be sensed by distal organs like the brain and in fact affect development or normal development, physiological development, or development of diseases. So people started to address that, and one potential mechanism through which this is happening is through the production of bacterial metabolites. So people show that some metabolites from the host microbiota can actually travel and reach the brain and affect the function of the microglia. And this microglia can actually control astrocyte response itself in response to these microbial metabolites. This is not happening only in steady-state condition, in healthy condition, like this group show, but also during inflammatory conditions. In fact, the microbiota of patients affected by multiple sclerosis has been taken and transferred in animal models which are genetically susceptible to develop the same disease in mice. And this was happening. So the feces of the multiple sclerosis patient were able to transmit the disease into mice that are at the beginning, they were perfectly healthy. So the microbes do something that even induce disease development. And people continue to work on that, and actually was shown later on by several different groups that what we described before, what we talked before, so gastrointestinal inflammation, it's linked and due to gut microbial dysbiosis or alteration in the microbial composition, and this induce abnormal behavior in mice. Big screenings, even at the human population level, show that people reporting signs of depression or low quality of life, they have they bear certain microbes in their intestine, or they lack certain microbes in their intestine. They could have a protective or adverse role on the central nervous system. The same experiment described before for multiple sclerosis patient has been performed also with autism spectrum disorder patients. In fact, transferring the microbiota from these patients into animal models was able to promote the generation of autism-like behavioral symptoms in mice too. So there is something that the microbes do or produce that can travel and reach the brain and affect the function. And we are really trying to understand how this circle happens also during early life. If perturbation of the maternal microbiota can actually induce perturbation of microbial derived metabolites that can pass the placenta, can even pass the blood-brain barrier in utero and reach the neuroimmune system and affect the neurodevelopment. So the open question of the project I'm studying, and it's funded by the PINC, is that how does the intestinal flora affect the central nervous system homeostasis, and which could be the pathways, the immunological pathways that are activated in response to bacterial exposure. And how could be this pathway mediate the central nervous system homeostasis. We started the project with the observation that if we colonize mice with germ-free mice, so mice that have never been exposed to any microbes, with only a single bacteria which is labeled with a 13C carbon, we are able to detect 13C labeled bacterial products or metabolite in all over the tissues of the mouse, and also including the brain, shortly upon gavage, 2 and 18 hours upon gavage. So there are microbial metabolites that penetrate the host tissues, including the central nervous system. And then we ask the question, how does the intestinal flora affect the metabolism of the host tissues, and in particular the central nervous system? And here we perform a metabolomic analysis of the brain of germ-free animals, so animals that have never been exposed to any microbes. They live in sterile plastic environments, plastic bubbles called isolators, versus specific pathogen-free mice, so mice that are colonized with a highly diverse microbiota, in which we don't know exactly what is there, but we know that some of the common rodent pathogens are not present. We performed this analysis and we actually identified several metabolites that are expressed in a different ways, either in germ-free condition or in colonized conditions. And then we thought, okay, these metabolites can be dependent on the presence of the microbiota, but can they be also dependent on immune pathway that they can be perhaps activated upon microbial exposure? And one of these pathways is the inflammasome pathway. Inflammasome is this complex of proteins formed by a sensor, an adapter molecule called ASC or piCAR, and an effector molecule called caspase-1. When this complex gets formed and get activated, caspase-1 is activated and it cleaves and releases major form of IL-1 beta and IL-18, which are pro-inflammatory cytokines. This complex gets formed and activated through two different signals, one coming from the exposure to microbes and the second coming from the cells itself. So then we investigated if animals which are lacking the inflammasome activation could show different metabolic profile in the brain. So here I present the second half of the graph that I presented before, where we included mice which are lacking the inflammasome. They lack piCAR or AST, the adapter molecule of the inflammasome. There's no formation of the complex in colonized condition or in germ-free condition. And as you can see here, in absence of the inflammasome, actually the mice look different than what a wild-type mouse colonized animals look like. So we have identified some metabolites which we are investigating now and try to understand how they can be mediated and regulated through the microbial exposure and the inflammasome activation. So our first hypothesis is that gut microbial signals such as metabolites are sensed by the host and shape the central nervous system and affect the central nervous system homeostasis. And we want to understand the role of inflammasome in these pathways, in these mechanisms. Of course, we want also to understand how this is happening early in life. So if the maternal microbiota or the microbiota that the mice encounter short after birth, so the early life microbiota, will impact the central nervous system homeostasis. And if the maternal intestinal inflammasome or the early life inflammasome activation in the pups have a role in that. So in order to do that, we are performing experiment where our mice colonized with a certain microbiota will are subjected to alteration of the microbiota through the usage of antibiotics during pregnancy or in the early life phases post-birth or constitutively throughout their life. We will analyze the brain function, the brain immune system, together with the intestinal immune system at the RNA level, protein level, and metabolite level to understand which are the pathways that are altering those conditions. So the impact of our plan, of our project, is actually to identify new biomarkers, new metabolites, and new mechanisms that are involved in the crosstalk between the microbiota and the gut-brain, within the gut-brain axis. And one of the potential mechanisms that can be involved in this is actually, in our opinion, our hypothesis, is the host inflammasome. Of course, the main goal is to understand how this basal and physiological pathways happen, to understand how we could then modify or manipulate what is happening during the development of a certain disease to reconstitute the physiological status. And one of our hypotheses that actually we could do that with the usage of bacteria as probiotics, or modifying the bacterial composition through the diet, or through genetic manipulation of microbes, or in certain cases, but in certain special cases, through the intervention with fecal microbiota transplantation. So with this, I would like to conclude my talk, to thank my two students, Andrina Ruch and Johan Kansolo, that are working with me on this project, my collaborators and colleagues from the University of Bern and from the University of Würzburg and Calgary, and the core facility in our institution that help us to perform metabolomic analysis and using animal models, and our collaborator for providing us the animals. And once again, a big thanks to the Bink Institute for funding this.